It's time for This Week in Virology, episode number 286, recorded on May 18th, 2014. Hey everybody, I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. We're coming to you live today from Boston, Massachusetts. We're at the general meeting of the American Society for Microbiology, a meeting that attracts thousands of microbiologists and is a really exciting place to do a TWiV. And joining me today, to my right, from Western Massachusetts, Alan Dove.
Great to be here.
Nice to have you next to me.
Yeah, out here in Eastern Massachusetts for a change.
Alan,
and a beautiful day for it.
It is gorgeous out there.
Gorgeous weather, lovely clear skies.
Let's see what the temperature is here.
Probably ought to check on that.
But we thank you for driving out here, Alan.
Oh, no problem. Traffic was easy.
It's not very far for you, right?
No, it's an hour and a half. It's not that big a deal. It's a small state.
Kathy Spindler happens to be right here at the meeting. It's 17 Celsius.
Okay.
Cloudy. She's right down the hall running a session. So there's a concurrent virus session going on at the same time, so she couldn't join us. But we do have a substantial audience here. And you do know there's a virus session going on here. Okay, but you'd rather be here. I can't blame you, really. It's more fun to watch us be silly than to watch PowerPoint presentations. So we have two guests today. We have pulled from the meeting. And on Alan's right, she's a professor of microbiology at the University of Texas Southwestern Medical Center in Dallas. Julie Pfeiffer.
Hi.
Welcome.
Thanks for having me and for the promotion, because I'm actually an associate professor.
Oh, we promote everyone.
Thank you for that. I don't have to go through the process.
Well, you're going to get the TWiV bump.
Okay.
So. And you'll be a professor.
You'll be a professor
When
soon.
you go home on Monday, you'll be a professor.
That'll be the first email you get.
Congratulations. Thanks for joining us. Been trying to get you on TWiV for a long time.
Yeah, it's
I was
great
supposed to visit, remember, and that fell through, but thanks for joining us. Also, to Julie's right, his second time on TWiV. He's a professor of microbiology, Boston University School of Medicine, and the director of cell and tissue imaging at the NEIDL, Paul Duprex.
Nice to be here, and for the promotion, but the advantage for me is my chair just happens to be sitting over there, so now he has no choice.
That's right. That's right. Why do you look like a motorcyclist today?
Why do I look like a motorcyclist?
You got the red arms, big muscles.
Well, I got the sunburn yesterday because the weather has just
Your
become
shirt sleeves
wonderful.
rolled up. You have a pack of cigarettes under there? as well?
Not a cigarette
Anyway,
inside.
thanks for coming back, Paul.
Yep, good to be back.
I gave you a bump too, is that right?
Mega bump.
All right, that's good. We don't have a problem with that. So we're going to, before we get into the science, we're going to talk a little bit about the needle, because we had some good news this week. But before we do that, I want to show you a T-shirt that Kathy Spindler gave to me just before the show. She got it from AAAS,
Speaking of needles.
Wow, you're
Nice.
good. Anyway,
I brought my A game.
It's got a picture of vials of polio vaccine and some angry-looking syringes there. And it's got a signature of Jonas Salk, I think, at the bottom. I was just very privileged to interview his son, Peter, for TWiV a couple of weeks ago, so you should check that out. On the back it says, "This is just great vaccines brought to you by science." That's right. Very cool. So thank you, Kathy. Poliomyelitis vaccine, which is here. This box is manufactured by Eli Lilly and Company, Indianapolis, Indiana, of course. And this is the 100th birth anniversary of Jonas Salk this year. So there's a big celebration. If you're in New York City in, I think it's October, they're having a big celebration. It's the virus you work on, right?
It is.
Oh, yeah. And I do as well, so it's pretty cool. So Paul, there's been some news about the needle lately. Last time I saw you, we did the movie about the NEIDL, which is a BSL-4 facility here in Boston.
Yep.
And
About a mile in this direction.
Tell us the hurdles it had to overgo to get to where we are today.
The short version.
The short version, yeah. Well, the two most recent hurdles that it had to overcome was an ordinance which a councillor from the city of Boston proposed that BSL-4 research in the city itself should not be allowed. That probably was the first and foremost hurdle.
Now, this is after it had passed several other reviews.
Yes, so it has gone through the risk assessments and secondary risk assessments, and Blue Panel and the NIH have awarded that grant. And the federal court have ruled, so all these different levels, but the two big hurdles was this city council ordinance, and secondly, the state court of Massachusetts also had to rule in favour of the facility opening. So those are the two final pieces of the jigsaw puzzle, so to speak. And this had been going on for a long, long time. The state court, we knew, had to decide, but the ordinance came a little bit, I guess, unexpectedly in the new year as an additional final hurdle to overcome. Well, amazingly, on Wednesday, those two dominoes fell one after the other in the space of an hour. The ordinance was rejected by a good vote.
It was 8-5
in the city council, yeah. So it's a really quite clear majority of the councillors looked at the evidence, listened to the NIH, listened to the Blue Ribbon panel, listened to really science, the fact that these facilities operate in many different places in the world safely, that there are a number of them in similar types of locations. In Atlanta, I've been in the one in Lyon. It's pretty much in the middle of the town. And they basically decided, yeah, we trust BU to operate this safely.
So they actually looked at other facilities and collected information. They didn't just make a gut-based reaction.
Well, actually, the council were very thoughtful, and they listened to BU. I think BU and also the opponents, the people who didn't want it in the city, they looked at it and they had a public meeting. Now, unfortunately, I was not able to attend that, but it was a 6-hour, 40-minute public meeting. Quite impressive.
Dealing entirely with the needle?
Dealing entirely with the needle. This is the councillors listening to the community, and BU is just one part of the City of Boston's community, so there's a community, everyone who has a vested interest in Boston.
Well,
So-
and there's a long tradition here in New England of having extensive public meetings.
It's really
about-
interesting. If you're not from this part of the world, which you might guess that I'm not.
Why would we think that?
Well, someone told me once I had an accent. Yeah, it's really interesting. It's very educational if you're from outside, what they do. And do you know what? It's not a bad thing at all. But I think what was pretty clear on Wednesday was the councillors had listened and they had looked at the evidence. And by looking at the evidence, the hard evidence, not just this knee-jerk, this is Hollywood, this is real, what happens on outbreak happens in Boston. You know, not that, because that's not the facts. They basically went with the facts, and it's pretty clear these can be operated safely in many, many places, and Boston is just a wonderful place to have it. Again, I'm not from here, but what do you know about Boston when you're not from here? Great universities, great pharmaceutical companies, small biotechs, a knowledge economy. You know, it's such a place that should have the, as Elke, who was on the last TWiV, Elke said it's like the Mercedes-Benz of BSL-4s. Whenever she gave her evidence to that council at City Council.
So you heard both sides during these presentations?
Well, no, I
is that
wasn't there.
right?
I was at the Society for General Microbiology annual meeting, so I had to deal with it by text. Ron was sending me a few texts in the middle of it, and it's still going on. It's still going on. It's still going on. So what I had to do is take a look at some of the video footage, and it really is quite interesting. I have not watched all 6 hours, 40 minutes of it. I can tell you that.
Well, we, of course, did a documentary of the needle, and everyone should go to TWiV and take a look at it. It's in the sidebar, Threading the Needle, which we did with you and Elka and Ron and our team from ASM. So I would like to think that helped a little, because we did tell people to look at it.
Yeah, and actually, to be fair, anyone, the needle leadership have done an awful lot with the community liaison committee, reaching out, educating, tours, anyone who wants to come and see the building within BU and within the greater community. They've brought people through it, and just in the same way, not to see all of the secondary space, but to actually see the lab space. So a lot of people have come through it and visited it, and I think that alleviates an awful lot of the concerns.
Sure.
And for the people who can't do that, you know, the people who live out in Western Massachusetts, it's not happening.
You know, since we made this documentary, I've been invited to 2 other BSL-4 facilities.
Yeah?
So I'm going to one in Australia this summer.
Oh, you're going to Linfa?
Yes.
Oh, cool.
It's, yeah, Geelong, right?
Yeah, in Geelong.
Geelong, sorry.
Oh,
And-
I have an accent, so...
And the other one is Rocky Mountain.
You're going to Rocky Mountain, very
Hamilton.
good.
So I'm going to do a podcast from both of them, so that'll be
Excellent.
fun. So thank you for the bump, right? So
But the thing which is pretty clear, and actually this is really interesting, and at ASM, I think it's important to say that whenever we are here, ASM sent, we had a petition. One of the things we wanted the City Council to see was that it's not really just a few people from Boston University. It's not just all this vested interest. There's a lot of people in the city, outside of the city, people connected with biotech who support this. And an ASM also sent that petition link out to their 37,000 members. Now I have to say, I've been a member now of ASM for, well, 15 years. And that really impressed me. That basically validated every single membership payment, because that's a society doing what a society should do. It's looking after microbiology. It's saying, you know what, this is important. It's not, okay, it's about Boston, but it's not just about Boston. It's not just about Cambridge, and it's not just about Newton and the other regions, and it's not just about Massachusetts. This is a national facility, but it's not just about the States. It's about all the other interconnectivity of science throughout the world. As you say, one in Australia that you'll visit, and one in Rocky Mountain, these other facilities. So I think I give an awful lot of credit. I guess this is a good opportunity to say that, to support a lot of the people who follow the tweets, and a lot of people who listen to TWiV, and a lot of people, our scientists, friends and colleagues really supported us, and we're grateful for that.
Could the council ever decide to bring this up again, or is that it?
I am not here, and I'm not knowledgeable enough of what they could do. I would say that would be unusual.
Well, there's an old saying that no one's life, liberty, or property are safe when the legislature is in session.
Yeah.
But it would be, once the facility opens,
Yeah.
it would become much more difficult to have
I
events like
already think that by having, even the ordinance itself was a little bit like trying to change the rules of Scrabble once you were halfway into the game.
It was kind of a Hail Mary pass.
It was very
unlikely
strange.
to have,
Yeah. So, but again, that's difficult. That's me looking as an outsider of understanding American politics, but which I guess not too many people from inside understand either. But I would be very surprised. It doesn't really make sense. I mean, who's going to... There's so many other issues that those councillors, in my opinion, should be dealing with in Boston. And NEIDL was probably an easy kicking bag at one point. now it's sorted. Just let's do the science safely, and let's really use that facility and make Boston Biomedical Sciences better than what it already is.
So when will it be open and functioning as a 4 facility?
Yeah, when do you start actually working?
Me? I don't work. I've never worked a day in my life.
You're sunburned right now.
Exactly. Why do you think I'm sunburned? There are 2 other permitting hurdles to overcome, but that's a regular system that all of those BSL 4 facilities have to go through. So the Boston Public Health Commission have to give it the AOK, as do CDC. And that can be anything from, I think the quickest I heard recently was 6 months, but that's not going to happen in 6 months. It can take anything from a year to, I guess, 2 years. You would like to think that in 2 years it would be operational. I just really would hope that that was the case. But it's a bit of a question, how long is a piece of string? Do you say that here?
I've heard it. I don't know if it's a local
We
thing.
say other things.
It's nice to teach you. It's nice to teach you people something.
Congratulations.
Yes, congratulations.
Congratulations, Ron, and everybody at the NEIDL.
Yeah, thank you. You guys played a big part in it too.
I don't think so, but
You did.
we'll take it.
Yeah, no, it did. It's in the same way as ASM, in the same way it's visibility, it's recognition, it's realizing that it's important. Yeah, I think microbiology is a community, and we live together, we die together, we support each other, and we should support each other as well. And I think it's really
Sort of a
really good.
biofilm.
Or microbiome, yeah.
It's a community.
It's a community.
It's a community.
Symbiotic, yeah. Okay, we've heard that.
It's good. It's
By
good.
the way, speaking of questions, anybody in the audience has a question, there's a mic here. You can come up. And also, those of you following online, you can send questions via Twitter. What is the hashtag? ASM 2014?
Yep.
Thank you very much. And we'll hear those as well. So become immortalized like a cell line. Okay.
All of your dreams.
That's right.
Let the healer begin.
Live forever.
Julie, let's pay attention to you a little bit.
Yeah.
Do you know Paul?
I do know Paul. We met in France, as many will, I guess.
Last at the European Society
For virology.
Virology in Lyon, the
Well,
meeting
that's
that
so-
you decided not to come to.
Yes. I'd had enough of you after the needle. Yeah.
You're
Try
not being
again,
invited
try again.
back.
Yes.
There was a good reason. Anyway, Julie, tell us a little. We know where Paul came from. From the last episode, 200, you gave us your history.
Did I?
From Belfast and all that.
Yeah.
So we don't want to hear it again.
Boring.
But Julie, where did you grow up?
Mine's far more boring than that.
No, you don't. Have you been to Belfast?
I have not.
It's boring.
I grew up in a small town in central Michigan. Belfast sounds more exciting.
Belfast is a good town.
It's called Midland, Michigan, 35,000 people, very flat. And it's where the world headquarters of Dow Chemical is based.
Oh.
And so for a very small town, they have a very high percentage of chemists and physicists and everything that goes along with that. So it was a great place to grow up and kind of grow in the presence of science and so
Your
forth.
parents worked for Dow?
My father did, yes.
Chemist?
Chemist. He can purify anything. Yes. Which I need to put him to work because now I
him,
have
yeah.
things to purify.
Yeah, bring them in the lab.
Yes.
Hey, Dad. can you-
HPLC. Yeah.
Nice. So that's where you got your love for science, probably?
Right, yeah. Watching him and going into his facility with him. But mostly I would say it was from a really amazing high school teacher named Sue Shiel. And we had a 2-hour block for an advanced placement kind of class. And we did a river study in the spring where we'd go out and sample every week. And we'd measure biochemical oxygen demand in the water and nitrates and all these things. And my job was to count plankton. And so I kind of feel like I've been a microbiologist since the age of 17 in a way. So that really inspired me. And so I became a microbiology undergraduate major because of that.
Where'd you go to undergrad?
Miami of Ohio. So it's kind of a liberal arts school. Small classes. I had a great time there. Worked in a lab that was great, Gary Jansen's lab. Learned how to clone, which served me well.
And at that point, you you took a lot of biology, and you wanted to be a
Right.
scientist already in college, or?
Right. I pretty much, I was more or less sure, not completely sure, but I mean, pretty much day one, I was a declared microbiology major. And I ended up taking a lot of classes that weren't required just because I found them interesting. Cell biology, things like that. I took an extra cell biology lab that was great.
So then you decided you would get a PhD.
Right. So I had done a couple of different research experiences. I did a NSF fellowship at the University of Iowa one summer. And that was the first time I had worked with viruses. And that was in Marty Stoltzfus' lab. He works on RSV. And I thought that was incredibly cool. And so I decided graduate school would probably be a good idea. And went to the University of Michigan and just had a phenomenal time there. I mean, it was a perfect match for me. And I found that I had this problem trying to decide between should I do bacteriology or virology as my thesis work? What lab should I pick? Because I was kind of torn between two. And in the end, virology won. But now much of my lab also works on bacteriology, ironically, so it all comes back.
All right. And whose lab did you work in?
I worked in Alice Telesnitsky's lab on retrovirus recombination on murine leukemia virus. And she started working on HIV around the time I was leaving the lab. But I was all strictly a model virus, model system, molecular genetics, those sorts of things, trying to understand mechanisms by which retroviruses recombine and aid genetic diversity.
So Alice was a postdoc in my department with Steve
Right,
Goff. I
of
saw her
course.
on a daily basis. I don't know, was she there at the time you were there, Alan?
I don't remember.
Okay. So it was-
You probably would have remembered her.
Okay.
She's got a lot of energy.
Okay.
So at that point, you were clearly on the track to being a virologist, right? So then a postdoc.
Postdoc came up, and I thought, it's a very weird thing picking a postdoc, in a way. And I thought, I want to switch viral systems a little bit and learn something a little bit new. I want it to be really tractable, but really I was shopping more for the mentor than the project, if that makes any sense.
Absolutely.
And so I interviewed in Carla Kierkegaard's lab, and you know Carla well. And I was just very impressed by the fact that she had projects in viral genetics, viral pathogenesis and immunology using mouse models. She was doing some pretty hardcore biochemistry with purified proteins and structure work. She was doing cell biology and autophagy, a few different viral systems going on, and I thought, that's a place where I can just learn. Lab meeting, you're just gonna absorb all kinds of different things every week. And that ended up being key. That was kind of everything.
Carla's brilliant.
Yes.
I've known her since I was a postdoc, because as I was leaving the Baltimore lab, she was coming in. I handed her my clone of the polio genome.
Which, thank you, because that's
You're
what we
welcome.
use.
Do you use the same
Yeah,
one?
it's the same one.
Yeah,
Vincent's given polio to everybody.
I have. Yes. But at the time, of course, this was was this before the eradication goal was?
So I
Set?
joined in 2001.
It's after.
And it was right around the time when everybody was, there was a decree, as if from the Pope, that you were to catalog your freezer stocks. And this is a big problem because a lot of people have fecal samples from kids in India, and they don't work on polio, but their freezer is full of polio, right? So it was this concern.
Or they have vials that are labeled Coxsackie virus that are
Right,
full of polio.
right.
Yes.
But I mean, people just studying Vibrio cholera or whatever, I mean, there's
Yeah.
polio in those stocks too. So it became a concern, and when I started in Carla's lab, I wanted to work on polio. I'm a huge fan of model systems. I kind of joke in the lab that I don't want to work on a virus if there isn't an infectious clone for it. So I really like the polio system. I like the power of it. I like what you could do, but I got a little nervous at that time because there was this feeling that perhaps funding for polio research might get reduced, even though you can use it to understand a lot of things. And so I did take on another viral system just in case, to kind of, as a backup.
What was that?
Hepatitis C virus.
Okay. So, yeah, I mean, and then eventually you started your lab, which is focused on polio.
Yeah,
So
initially, yeah,
you didn't worry about the eradication and how it would affect research.
I worried about it. I mean, I guess it didn't
Yeah.
stopped me so much. I mean, I guess I was really interested in the power of the model and the power of the genetic systems and just really interesting questions that in spite of 100 years of research, we still didn't understand. We still don't totally understand why did so few people become paralyzed. Even in the pre-vaccine era, less than 1% of people that were infected became paralyzed. And why was that? What limits dissemination in most people? You know, most people never even knew they had it. So what's good in them and bad in others?
Right.
And I thought that was a great model to be able to understand how a lot of neurotropic viruses move around the body and so forth. So I very much started my lab on polio and Hep C. My very first student, her whole thesis was hepatitis C virus on a completely unrelated project, because I was a little bit worried about having all my eggs in one basket. But the polio stuff panned out, and it ends up, the things that we're learning with polio are applicable to a lot of other viruses, and so people, I think, are a little bit more willing to listen now, perhaps. And we've also taken on a lot of new viral systems in the lab. We have reovirus, Coxsackie virus, Tyler's, EMCV, yellow fever. I think
Also, if funding becomes tight for polio, it's not as if that's a unique problem in the polio field.
right, that's a problem for everybody, yes.
So this idea of model systems is, I mean, the appeal of model systems has gone up and down over the years. At one point they were loved, and then there was a phase where, you know, if you wrote a grant application and used the word model systems, it would be a problem because the idea was to work on relevant pathogens. But now we're coming back to where model systems like polio are valuable. And
I hope
saw
so.
that. I saw that in all the years of applications. It was harder at a time when, because polio is a model system.
Right.
You know, almost eradicated, it's hard to say the research is good for anything else, although there are some post-eradication applications, of course. But it's a great model system. In fact, I have used your papers, both as a postdoc with Carla and from your lab. I use them in my courses all the time because they're great examples of how studying a model system can illuminate all of viral problems. So I want to talk about this cool observation that you made, where you say your interest in bacteria and virology came together. The idea that the microbiome of the intestine is involved with polio replication. Maybe you could tell us how you got interested in that.
Right, so that was an interesting circuitous route to something that became really, really interesting. So this was all started with a graduate student, Sharon Coos, who is a TWiV listener. So Sharon, if you're out there in the universe. And she was my second student in the lab, and she was interested in looking at viral dissemination and host barriers that limit viral movement. And in her first study, she found that the gut was a major barrier that impeded poliovirus dissemination out of the gut that limited its dissemination to the central nervous system. And so we became interested in the gut as a barrier and what might be going on. And so we came up with this hypothesis, you know, that the microbiome is so big, you know, the last 10 years or so. We thought, I wonder if bacteria in the gut are somehow protecting the host, because they're known to protect the host from, you know, salmonella infection, you know, these pathogenic bacterial infections. And we wondered maybe if something similar was going on with viruses. So we kind of had this hypothesis, you know, you jot down all the hypotheses that you come up with, and then you kind of pick which ones to prioritize. And I would say that if it hadn't been for a specific colleague, that hypothesis would have been very low on the priority list, because I'm not a gut person. You know, I had a little bit of training in bacteriology, but not very much. And the gut is a very scary place. I mean, there's a lot going on in there. So the entire reason we started this project is my colleague Laura Hooper, who spoke, was that last night? Last night. She is in our immunology department. She's joint appointed in our department. And basically she's my best friend, and we drink together twice a week. She comes over and we have drinks and dinner on my patio, and we go out with friends Friday nights. But we talk about science and other things on this patio, and we talk about ideas for projects, and there have been a lot of projects born on that patio, including one of hers. We were just talking about, I wonder if circadian rhythms in the gut do anything. And then, you know, flash forward 2.5 years, it's a science paper, you know, and we followed the course of this project. So this was very much one of those times where just talking to a colleague that works on something a little bit different from you can be really good. So she said, interesting hypothesis. send Sharon up to my lab. She can talk to my students and postdocs. They'll set her up. We can tell her what antibiotics to use. And so that's how it was born, was just out of that. And it ended up being that we were totally and completely wrong. Our hypothesis could not have been more wrong, which I find more exciting, actually. It's kind of fun when biology is smarter than you are, which is always. So Sharon found that if she depleted gut microbes by giving the mice antibiotics, that the virus no longer replicated and then the mice survived better. And we were expecting the exact opposite result. And so then it became this interesting phenomenology. And it took us a long time to get some traction on it. There were a lot of twists and turns. We were misled a lot. You could have a whole episode with Sharon talking about her misspent year. But in the end, trying to figure out what's going on, how is this all working, ends up being the big challenge. And we're still working on that now.
So which mouse background do you use? What's the
genetic background. So these mice, normal mice are not susceptible to polio.
So these are transgenic
These are
animals?
transgenic mice that express the human polio receptor, which he has generated
These are
somehow.
Rebow's mice, right?
Yeah.
Well, actually,
Probably not,
Right,
because
because that one, yes, got locked
Columbia
up by
wouldn't
Columbia's
let me give
stupid
them to
patent
anyone.
office,
Yes.
yes. Anybody from the Columbia Patent Office who's listening, that was for you. Yeah.
I didn't ask that question for that reason. I just wanted to know
Yeah.
which
No,
mouse.
no, yes.
we're just looking to
well,
complain.
and they're also
Sorry, we digressed a little bit there, and
Sorry, Jim.
that's personal.
politics.
So you use someone else's transgenic.
We use Satoshi koikes because they also lack the interferon alpha beta receptor. So they're immune suppressed, and then they have the polio receptor, and so they're orally susceptible
Right.
that way. That's the only way to get them
Right.
orally susceptible.
Wild-type mice, even if you have the receptor and you feed virus, it doesn't replicate? Is that correct?
Not well at all. And that was a big problem in the field for, probably 10 years. You and others tried to
Right.
get polio to, infect mice by the oral
Yeah.
route, and people went to heroic efforts. And
It was Satoshi Koike in Tokyo who figured out that if you take away the type 1 interferon receptor, then the virus will actually replicate in most tissues wherever there's a receptor.
right,
So those are the animals you use for your
that's
studies.
right. Yeah, we use the immune competent versions as well as a comparison. It turns out the virus replicates a little bit. It replicates kind of okay in the gut of immune competent mice, but it doesn't disseminate out of the gut. So it's a post-gut block that interferon is doing.
So when you depleted the gut microbiota, then the virus replicated to lower levels?
Right. It basically hardly replicated at all. And the mice survived much better, as you might predict. And so then the big question becomes, why is that happening? And we're still pushing on because we've identified some mechanisms that might be going on, but it may not be the whole story.
Sure.
So one mechanism that we've been characterizing are effects on the viral particles themselves. So there are a lot of labs looking at the effect of microbiota on innate and adaptive immune responses that then can have effects on viral, replication. We started by looking at viral particles. And what we found is that bacterial surface polysaccharides, including LPS, which is on gram negatives, or peptidoglycan, which is on gram positives, both of those polysaccharides will bind to the viral particle. And we figured out a lot about the requirements for what the polysaccharide needs, but both of these things work. Binds the viral particle, and it does two things. The first thing is it aids the virus attachment to its receptor, which obviously might assist its replication process. And the second thing...
Where is that binding in the capsid? Because there's a pocket for...
Yeah, antiviral drugs,
Right.
lipids.
Right. The truth is we don't know the exact binding site. We actually have a mutant that binds less, but it's a conditional mutant, so it's a little bit complicated. We think the binding site is probably nearby.
Okay.
This particular mutant's in VP1, near the 5-fold. For those aficionados
Yeah,
that know what that means, sorry. Talking
A little
shop.
inside there.
Oh,
Yeah.
we shouldn't talk about viruses on
No,
this
we
week
shouldn't
in
talk about
virology.
viruses. Yeah, we should talk at least at a level that other people can understand. There's a history of odd molecules binding to
Yes.
the virus. of
So the
polio.
receptor binds around the five-fold
That's right.
axis.
Right.
In fact, you can see a picture of it here on my
Yes,
phone.
on the TWiV logo, as a matter of fact.
This is polio binding its receptor. You can see the five-fold axis of symmetry. So the question is, does LPS bind in the same?
Bind around in that.
It's around there,
Around that.
but not that exact site, which would
Okay.
make sense.
Right.
Yeah, so it aids receptor binding. And the second thing that it does is it stabilizes viral particles, and it makes them resistant to heat, and it makes them resistant to dilute bleach. Very dilute, short period of time. We can definitely kill it, don't worry.
From what temperature?
It'll survive 42 degrees, 6 hours, no problem. We've gone much higher than that. It'll survive
And without it, what?
dead in less than an hour.
Interesting.
Yeah. So what we're trying to
So
do
it's
now is-
stabilizing the particle and helping receptor binding.
Right. So the stability, we think, that doesn't matter so much for the actual infection of the primary host because you ingest polio and it's going to replicate within a couple hours at body temperature. That's
Right.
no problem at all for the virus. We think it's more important for transmission. So viruses have to be stable in the environment. So for, as gross as it is, fecal orally transmitted pathogens, they need to be stable in the environment long enough to be picked up by that next host. And so we have this mutant virus that doesn't bind quite as well and it's not stabilized, and we can show that it has a transmission defect in mice if we age the feces out before we use it for the second.
You age the feces, like
Oh,
steak,
yes.
right?
Like wine.
Like wine.
Yes. We do. We age. It's a I believe a 4-day room temperature.
Your lab must
Cure.
smell great.
It's down the animal room.
No worse than any animal
So,
room.
but in the mouse, LPS and other surface material are important for replicating in the mice. So the transmission
Right.
issue is something else.
Transmission issue is something else. So that primary cycle in the host, the receptor thing might have something to do with it, but there are probably other effects, things having to do with innate immune priming. And in fact, there are a lot of other groups working on this, and they have really good evidence. A great example is Tanya Golovkina's work. So our papers were published back to back, which was great for both of us, obviously. And she works on a mouse retrovirus called MMTV, totally unrelated to polio. And she found that it too binds LPS, and that the virions that are bound to LPS initiate a very specific kind of innate immune signaling that culminates in IL-10 mediated immune tolerance. So it allows the virus to keep replicating under the radar, essentially.
Now, LPS is not specific to any particular microbiota.
Gram negatives, and there's a wide array of LPS types and the kinds of glycans that are
That'll do this.
right.
So soil microbes would probably be
Right.
just as good for polio as,
Right. It's an interesting question, actually. Right now, Chris Robinson in the lab is screening through. We basically went to the Med Micro Lab, where they have like every bacterium you can imagine, and said, yes, we'll take all of these. And so he's screening for activity against poliovirus and a bunch of other picornaviruses, Coxsackie B3, Tyler's, EMCV, Rhino, to see if do they all work, you know. So it's kind of the
So
pan.
and rhinoviruses would be a tract pathogen.
Right.
I'm sure there's a microbiota of the mucosa as well, right?
Right, there is, but it's not nearly the numbers in the gut,
Yeah.
right? So rhinovirus, we might expect might behave differently. We don't know. It's just kind of interesting to screen.
So right
through.
now, you just know polio virus it has this property in the gut.
Well, and Coxsackie B3 also.
And do you have any way of knowing whether this is the same in people? Because everything's in
Right.
mice so far, right?
Yes, we have no way of knowing in people. People have asked, well, have you looked back in the old literature to see if there's any correlation between antibiotic use at time of a, you know,
polio infection.
endemic, yeah, and I wouldn't even know how to start something like that. And the truth is, I wouldn't really expect there to be an effect because Sharon did some experiments so we used 4 antibiotics at very high concentrations that would never be achievable in a human. So we really knocked down their flora. And it turns out if one of the antibiotics went bad without her knowing, and in that experiment, instead of getting a 1,000,000-fold knockdown of bacterial count, she only got 1,000-fold. And then polio was perfectly happy. to replicate.
I think you need to get human feces clearly and do the experiment.
Aged.
Do you want to do a sabbatical
Yeah, I'll do a sabbatical. If I can sit in on your,
on the patio?
Yeah, the patio.
Oh, yeah. If you're willing to age human feces.
yeah, no problem. That's fine.
It's a deal.
He'll leave them on the patio for you.
So basically,
I knew it would be as bad as this. I knew this would happen. It's just
I kind of have-
inevitable.
I have a
It'll always go
reputation.
there.
I'm not going to say anything there.
It's a family show, by the way.
don't.
so-
So basically, the virus has evolved over time to be able to benefit by stabilization or some other thing in the gut, right? It makes perfect sense. You might as well use the community.
Well, that's the whole
there.
thing about evolution with viruses. So RNA viruses, as polio is, they evolve a millionfold faster than we do, than bacteria do. So they're not, it just makes sense, and they undergo replication so quickly that there is such selective pressure on these populations that if one has even a tiny advantage, it will very quickly take over the whole population and benefit from what's there. So it makes sense that the viruses have evolved to
Yep.
use what's there.
I make this statement in my evolution lecture. So humans and chimps differ by 2% of their DNA. It took 1 million years to achieve that. Polio achieves it going from the mouth to the intestine. So imagine what polio could do in 1 million years.
That's right.
So I view it, earlier you talked about this, this question of why the virus doesn't efficiently get into the CNS. I view it as an accident. I think this is a exquisitely adapted human virus to the gut.
Agree
Right.
completely.
I mean, most people, you get infection, you don't even know you have an infection. The virus replicates for weeks, very efficiently, it's spread, and you're fine. But maybe, and then 1 in 100, 1 in 200, it gets into the CNS. I think it's an accident because there's no good to a virus to getting in
It's a
there.
total accident and it's a dead end.
Right.
I mean, what's the benefit? You're not going to be transmitted from the spinal cord,
Yes,
right?
unless you're an animal eating another.
Well, that's what I've
right?
thought about before is maybe precursors of poliovirus that were not human specific. If there would be some predator-prey thing where eating
A paralyzed mouse would give it to the cat, yeah.
something like that, but I mean, why would it be maintained for polio when it confers no benefit?
Yeah.
Right.
Yeah, right. We actually don't even know where polio evolved from. Unlike many other viruses where we kind of, your virus, measles, right? We sort of know.
Well, I mean, it really is interesting listening to you because it's very, what you're saying for polio, there are so many nice parallels with measles. Measles is a dead end in central nervous system. In fact, it's an accident that it ends up in the CNS. But that's what I was just going to ask. Where do people think polio came from? Because ultimately, what we imagine is they're coming from animal reservoirs at some point.
Sure.
So we're so human focused
Yeah.
with our viruses because
Well, and
when you're not working with model systems, what are you working with? You're working with viruses.
And there are
that
plenty of other intestinal viruses and even picornaviruses that never have any neuropathogenesis. So
That's right.
why is it that polio
Great
has this?
question,
Maybe there's something about its particular evolutionary path that has left it with this unfortunate side effect, or maybe there was some advantage.
We have no
at some point in the
idea
past.
where polio originated from.
So
So
you
your
say-
virus
Right.
probably came from rinderpest.
Well, that probably is it's who knows?
It's an idea, but we
It's
don't
an
even have an
It
idea
came from
for
some
polio.
sort of
Because there are no polio-like viruses out there that we have discovered. Maybe we have yet to discover them. We need to go out and, look for picorna-like viruses in the wild, but we have no clue where it came from, so we can't even speculate about why it would have been in the CNS at all.
But you see, if you see something like Nipah virus,
Nipah, yeah?
Nipah, which could be because it is jumping from bats into people and bats into pigs, and many more central nervous system complications there. So what measles is now, very, very tuned exquisitively to people, just like polio, exquisitively tuned to people. That's why the animal model's so hard to make.
Yeah.
Nipah and measles could have been much more similar whenever you look back in evolutionary history, whenever measles, whatever it came from, was initially jumping out of that animal species.
Yeah. I always like to compare polio and, say, norovirus, where, so polio gets in, it replicates. Most of the time, there's no symptoms, and it efficiently spreads nonetheless,
Right.
without being pathogenic. And norovirus, as you know, bam, you get infected and woo, 2 buckets.
Also efficiently
Right.
spreads.
and it efficiently
But,
spreads.
but
it-
there, the difference is the pathogenesis there aids transmission.
Yes.
If you can aerosolize feces, transmission.
I understand, but you don't have to.
But why didn't polio ever?
Sorry.
Yes.
I'm so glad I work with
Go
simple
try that at
viruses.
home. Yes.
Simple respiratory viruses. Hopefully people aren't eating during this.
It's ASM, they don't care.
I'm thinking about the people at
Will that
home.
be part of my sabbatical work?
Whatever you want to do, I'm happy to accommodate. If you
But-
write the safety plan.
So you're right, aerosolization is a great way, it spreads on airplanes like that.
Right, so why didn't other
Exactly,
coronaviruses pick up
so you
on
don't
it?
have
Yeah.
to, well, the answer is that whatever path to transmission you achieve when you evolve works. There doesn't have to be just one path, I suppose. But neuro is quite virulent, right?
Yeah.
And being virulent is a good way to achieve transmission, either by vomiting, diarrhea, coughing and sneezing. But there are other ways too, I suppose. That's the only way I can really
And also, each of these
describe.
viruses got to its current point through some path, which has left it with molecules that may not be ideal for its current lifestyle.
Yeah.
So it's constrained. Polio, particularly with the tiny genome and tiny capsid, it's very constrained in what it can change, and maybe it could change something to get around neuropathogenesis, but you can't get there from here.
Right.
That path has been lost to it.
It's very interesting that we have had to immunize globally against an accidental infection of the CNS, right?
Right.
But of course, that's what we see. Paralysis is a serious consequence. So now, you're trying to sort out the details of why, it's not just LPS, but there are other things you're trying to sort that out as well?
Yeah, so we're trying to figure out, what are the glycans that can do this, and do some do it better than others, and trying to hone in more on the specific molecular mechanism of, right now we're really focused on stabilization. And we have this new hypothesis that we've been testing, and this is the idea that since each viral particle has 60 capsid proteins, there are 60 potential binding sites
Right.
per variant. And we know that for these polysaccharides to have activity to stabilize the virus, they have to be long. So we can use little 6-unit long oligomers, and those don't work, but the longer ones do. And so one hypothesis we thought of was, what if they're clumping viruses because they're binding the same, polysaccharides? And it got me looking back into the literature, and some of my favorite things to do are to look in papers from the 60s and 70s, because there's such good stuff there that my student just gave her presentation last week, and she showed a picture from a paper from 1961, I think it was. It was great.
You
But-
weren't born at the time, but I remember being alive. Neither were you.
No, I wasn't around in
Neither were
61.
you. Oh,
Nope.
man, Jesus. Okay, sorry.
So we have this hypothesis that maybe these things are aggregating, and if you look in the literature, people have seen viruses aggregate in labs.
Yeah,
I mean, this is
sure.
a big thing that in labs you don't want your viruses preps to aggregate because it screws up the plaque assays and all these things, and you don't know what you're looking at. But everybody fights this problem. And mainly in the 70s, people were concerned about it in sewage, getting back to my favorite topic, in wastewater, because viruses can aggregate in wastewater under certain conditions, and they become very, very resistant to inactivation. So that's obviously a problem for wastewater treatment. And so there were all these papers looking at what induces viral aggregation and the fact that they're hyper-stabilized, and I thought, well, that kind of sounds familiar. So we've been looking at this hypothesis that maybe these polysaccharides are inducing viral clumping, and that those clumps have enhanced stability, and maybe the clumps are the transmitted form.
Right.
And thinking about the genetic implications of that. So we've done several different assays to look at that. We've done electron microscopy, and we can see clumps when we add LPS, and they're not there otherwise. They're not huge. They're maybe 10 virions per clump. We can do things like dynamic light scattering, which is a biophysical assay where you can measure particle size by, how light scatters in a solution. And there we see increased size in the presence of these polysaccharides. And then we have a genetic assay, which is my favorite one. My background's genetics. And so we had made 10 genetically tagged viruses that we used for a lot of different things in the lab. And we're all taught that a single bacterial colony or a single viral plaque was initiated by a single founder landing at that place on the plate. But what if a clump initiates that event? And so by taking those 10 genetically tagged viruses, we can ask, does a single plaque have more than one tagged virus in it? And the answer is yes, in the presence of those aggregation inducers. And because our assay for that just relies on plaques and RT-PCR, we can do it in vivo. So we orally inoculate mice, either antibiotic treated or not. And then we take their feces and we generate plaques and we ask, okay, how many of these plaques have more than one virus in them? And it's higher in the mice that have their microbes intact. And so we're thinking a lot now in terms of, is that the transmitted form? Does that aid transmission? What does that do for evolution? Because we know that if you have more than one genome in a plaque that you can detect, it means that more than one virus was actively able to initiate replication. And so we've been thinking a lot about complementation, recombination.
It's nearly like polyploidy. It's a way that a virus which has a non-envelope virus can be polyploid because the capsid makes it so constrained that it couldn't possibly encapsulate two genomes.
Right.
But it can basically bring its brothers along with it and become.
Well, it kind of almost is like segmented genomes, right? But with individual.
In bits.
Yeah.
Bring in extra copies in case one is,
It's like a retrovirus.
Exactly,
because
they're diploid.
Well, that could really stone a couple of birds. You've got the particle to PFU problem,
Right.
which polio has a lot of particles that aren't going to productively
That's
infect,
right.
but they could be in these clumps and bring along an extra copy of the genome if that's the whole entry unit.
Right.
And you could complement mutations because it's a very error-prone RNA virus.
It's almost like you wrote my grant. It's amazing. Right.
idea.
So the idea is these viruses have roughly one error per genome.
My services are available for hire.
I would pay. For polio and most RNA viruses, they have roughly one error per genome, and that's going to inactivate a lot of the particles. But if you have two viruses with different genome defects that undergo synchronous co-infection because they're forced to through aggregation, you could get complementation of those defective proteins, facilitate a couple cycles of replication, they could recombine. And the beauty of polio is we have all the genetic markers to test that. So we have temperature-sensitive markers, drug-resistant markers. And then we're also really curious about your idea with segmented viruses. So this whole idea of genome reassortment. So the whole reason you have to get a different flu shot every year is that out in nature, in birds and swine, these viruses have multiple genome segments, and a cell can get co-infected with more than one virus, and then they randomly reassort, and you get a unique, novel variant. That if it has a selective advantage, it grows out. And I've always thought, statistically, these are incredibly rare events that a single pig or duck would be co-infected with two different viruses at the same time, and the exact same cell would be co-infected at the same time. But what if there was some clumping mechanism? Maybe that would facilitate synchronous co-infection. And so to test that, we're collaborating with Terry Dermody's lab on reovirus, which has segmented genome. And again, great model system, genetic markers, temperature-sensitive, drug-resistant. So we'll see if the frequency of genome reassortment increases if we can clump the viruses and so forth.
In your same polio paper, didn't you also show that Rio was dependent on the gut?
Right. That was a part of Sharon's original paper in collaboration with Terry's lab. We showed that Rio also relies on the microbiota for replication pathogenesis.
You sound very much like Carla, the way you adore genetics. And if I remember, she also loved to look at the old literature as well.
Yeah, that's true. She did.
I remember her quoting a paper of George Palade from 1960 where they saw vesicles in polio-infected cells, which today we think are where the replication occurs.
Right.
So you learned well from
By osmosed.
Yes. We have a tweet here from Layla who said, I'm watching this and it is wonderful. I love Dr. Pfeiffer. I want to hang out on her patio.
Laura's going to kill me.
Going to get all these people coming. Well,
How big is this patio?
yeah, that's right. Hey, Paul, can we talk about measles and canine distemper virus a bit? Yes?
Paramyxoviruses.
I thought you didn't do any work.
I don't.
I was looking at your papers in preparation for this, and when I searched for Duprex P, I found nothing.
Yep.
because I have to search for WP. What's the W?
So you can blame my mother on that. She'll be very impressed that I've now mentioned her on TWiV, because she might even be watching this in Ireland. So hello, Mom. Has that ever happened on TWiV before?
I don't think we've had anybody
Your mom
say
could
hi to
tweet
Mom,
you.
though.
Yeah, she can
remember,
text. She
probably.
can text me right now if she heard that. My mom decided that William Paul sounded better than Paul William.
So you're actually
I don't
William
know if you
Paul.
agree. Do you agree?
William Paul.
I like W. Paul.
W. Paul.
W. Paul sound. Yeah, that could be.
Why don't you like William though? No,
She never called me William.
She called you
I was
Paul?
always called Paul. She
Okay.
likes the sound of William Paul, but she wanted to call me Paul. And I guess that's a mother's prerogative. I didn't have enough
Okay.
of that. I didn't have enough of that to say about it at the time.
So if you want to find Paul's paper, you have to search for Duprex WP.
Well, actually, you don't.
You don't have to
You
search
just
at
have
all.
to search for Duprex
Duprex?
because it's
Maybe
unique.
that's
it's
right.
unique. Yeah.
I automatically did Paul because I figured there were so many
it's,
Duprex in the world.
Duprex is very findable
Okay,
on PubMed.
I'm going to do it right now.
I put
DUPRES.
Duprex virus.
Duprex virus. Oh yeah, there
That
they
works.
are.
Okay.
Okay, so
They published in
anyway, don't
viruses.
use P. That's just a side thing, which we never do on TWiV, of course.
No, we never digress.
So I wanted to ask you about both
How'd the stump grinder work out?
the stump grinder. The stumps are still there.
Oh,
Actually.
well, you haven't hired him yet.
No, they're
Oh.
still there. Every time I cut the grass, I have to go around the stumps. It's really
This is the second arc.
Excuse me?
Second arc.
Stump grinders?
Stump grinders, needle.
Oh, do we only have 2 arcs?
Well,
oh, no,
two
we're full
in
of
one
arcs.
episode's pretty good.
We've got arc lamps here, too. No.
One
I'm
ton
sorry.
too many.
You were talking about his papers.
don't remember what I was about
You were talking
to
about
ask him.
PubMed and
I've got papers.
Paul's papers
So
come up. William Paul's papers come
You
up.
study measles. Let's start with measles, and I want to talk a little bit about canine distemper
Yep.
virus. Measles you study in macaques, is this correct?
Well, actually, it's interesting listening to you talk about polio, because the very first in vivo work that we did with measles was with an alpha beta interferon knockout mouse, which expressed a receptor, a transgenic animal,
Yeah.
which expressed CD46. And that's an interesting connection back to ARC, back to what you were just talking about, because we were working with mice, because the only animal model that you can use.
Mice.
Yeah, the only proper natural infection animal model that you can do for measles is macaque.
Okay.
But that's very expensive, and you have to collect an awful lot of data before you're able to convince anyone to look at the viruses that you've generated in such, and ethically as well. You have to really know what you're doing. So we started basically being interested in measles neurovirulence. And it wasn't even a simple question of feeding them the virus. We injected it IC and then looked to see how the virus spread within the CNS. And that was at a time that we didn't even really know what the proper receptor for measles was. So this CD46 was a cell-associated in vitro artifact. It's not the actual receptor for the virus at all. So from that small animal model of measles, we wanted to do something more natural. And again, this was in Belfast. We then were working with canine distemper virus. And canine distemper virus is really a very closely related cousin of measles. It has an awful lot of the similar attributes of the infection of measles. Measles is incredibly lymphotropic. It's epitheliotropic, and it's also a little bit neurotropic.
I like this part.
A little bit neurotropic.
Little bit neurotropic. Whereas CDV is like a souped-up measles. It's very lymphotropic. It's much more epitheliotropic, and it's very neurovirulent.
And CDV is a pathogen of dogs.
And
right?
CDV is, it's called canine distemper virus, but you really could call it carnivore distemper virus, because CDV, again, unlike measles is a long way down that evolutionary route. It's exquisitely adapted to primates. CDV is not quite as far along that, so it infects a wide range carnivores, not just carnivores, but a large number of different wildlife species in the States, for example, can be infected with it. Fisher cats. In the States, the raccoon population really replicate. There's massive numbers of raccoons, millions of raccoons in this country.
We have raccoons on my patio.
We have a raccoon. We have a raccoon in our back. Our trash cans just are in the garden.
I actually saw one when I was living in New York City. I saw
They're
one outside my
unbelievable.
apartment building running around under cars once.
Yeah, I had not seen them until
Of course,
I
New
lived.
Yorkers
we don't
thought it
have
was a
them.
giant rat.
Yeah.
No, come on, it's
They
a
live-
raccoon. It's got a ring tail and everything.
They live in Central Park.
So anyway, the raccoon get it. But the interesting thing about CDV, it jumps into many different species. So, for example, lions in the Serengeti. Yeah. There was a very interesting, rather sad outbreak in Texas in the States. A big animal sanctuary lost, I think, 8 or 9 of their big cats that last year because of CDV.
Is it because it's neurotropic? That's
Well,
why it's lethal?
so CDV is also the other hallmark of these viruses is they're really immunosuppressive, so they really infect massive numbers of immune cells.
I see.
And then, again, linking back to what you're saying, this interplay between viruses and other infectious agents, viruses with bacteria, that allows secondary infections to take over, and that's now jumping back to measles. That's what leads to the death of the kids in Africa who don't get vaccinated or who get exposed to measles before they're able to be vaccinated. These secondary infections are really very ready to jump in, pneumonia with the kids because of the immunosuppressive nature of the viruses. So back to the original question, starting with a mouse for measles can't do the primate model. What else do you look for? If you're really interested in pathogenesis, which my lab is, the CDV in a small tractable animal model, which is the ferret, is a really very good way to study souped up measles. So
What did you say? Souped up?
Souped up
Souped up.
measles.
Is that a word?
Yeah.
Sure.
I
Oh, yeah.
In this country,
I'm just trying to figure out what it means for measles. Souped up?
Well, CDV
Pimped
is
out.
souped up measles, right?
Okay,
So
CDV. Got
it's
it.
like measles on steroids,
Got it, okay.
except you wouldn't treat measles with steroids. So souped up measles is a better,
souped up measles.
All right.
So, it's really interesting. So you can take the CDV and you can infect the ferrets with it. Simple intranasal inoculation.
Does it spread by aerosol?
Oh,
between
CDV,
ferrets?
absolutely. Yeah, really very. That's the other thing about these morbilliviruses. They are, now that smallpox is gone, these viruses are the most infectious human, measles is the most infectious human pathogen. The R0 is 12 to 15. Very infectious, which is why whenever people don't get vaccinated, you have an outbreak
You get
in
an
New
outbreak.
York.
Yep.
Was there an outbreak in? No, it was just outside Boston. We had
Right.
a couple of people in Trader Joe's.
These are people who are not immunized, correct?
Yeah. And there's a big outbreak in the Philippines at the minute, and an awful lot of cases are coming into Ohio and California, San Francisco.
It's mostly in communities that are not immunized,
Yeah.
and then of course the vaccine is like 99% effective, so you've got 1% of the
After
population.
two goes,
that could-
Alan. After two
Right.
shots as well.
So there's some tiny percentage of the vaccinated population that's also susceptible, but it's mostly you get the nucleus of the thing going in a place where you have an unvaccinated population.
So back to CDV.
So back to CDV.
Souped up
This
measles.
Souped
is
up
really
measles.
very hard. Does anyone follow this?
They can rewind. See, it's live streamed, but they know they'll be able to back it up.
So we did the infection with the CDV, and the other thing that we have really taken a back step with is we used a lot of lab-adapted viruses in the past. That's how I started. That's how I set the reverse genetic system up for measles using Martin Billiter's system from Zurich. And these are viruses which are joined together from all bits and pieces, scraps of this and scraps of that. They really don't represent proper clinical isolates. So we took a little bit of a back step with the CDV work and took an isolate that we really knew the provenance of, and knew that it caused disease in the ferrets, and then took it and made a version of it which expresses green fluorescent protein, which allowed us to really understand pathogenesis in fundamentally different ways, because you literally can watch the virus spread from cell to cell to cell to cell in the same animal over time as you bleed it day on day. And then at the time of necropsy, whenever you're able to shine light on the animal, you really get to see, because of the fluorescence, where the virus is, and that allows you to do very, very targeted pathology. So instead of looking through this whole animal for the tiny bit of replication here and here and here, you can really focus straight in. Right. You can isolate that piece of tissue, you can block that piece of tissue, and then do the pathology on it. So I think by collecting the data in the mice, this is why these transgenic, you really need to do that, and then going on to the ferrets and developing those types of approaches. We then hooked it. My lab has never worked with macaques in Belfast. We just didn't have that ability. But I have a colleague, a friend in Erasmus Medical Centre, Rick de Schwart, and we met in the European Measles meeting. So there's a small group of us got together each year in Germany. And whenever he saw what we were presenting and saw the viruses, and we chatted, and he said, we really should do this experiment in macaque. And he's an exceptional scientist, incredibly meticulous, very careful, and has invested a huge amount of time in that model, and does it very, very well. Has a great team of people there as well, that he's really at the forefront. And then we took one of the viruses, and we did that natural infection of the macaque.
So natural meaning respiratory?
Yeah, so the guys in Rotterdam have established that model with an intratracheal delivery. And the reason why they do intratracheal is because you can really be absolutely certain where the virus is going. So the very first time we did it, and plus as well, you want to make sure, if you're putting a small amount of virus in, you want to make sure that the animals are actually going to be infected. You don't want anything. These are very expensive. Again, there's ethical considerations to setting up and to doing such an experiment. So you really want to make sure, if you're going to ask that question, can we really understand measles pathogenesis in the optimal animal model, we better make sure that those animals are going to be infected.
But in a natural infection, you don't get intratracheally inoculated.
Absolutely,
right?
but
it infects the upper tract first, I presume?
No.
You inhale it deeply?
Yeah, so this is what we wanted to ask the question, but before you can ask that question, you want to make sure that the macaques are infected. You don't want to go in and infect 50 macaques with all these different routes and all these different amounts until you know that you can, using the gold standard route, that you can infect them, that you can understand when the peak of infection is, when the early infection is, when the virus is cleared, and then use that knowledge to really ask the question. And again, it's back to understanding primary pathogenesis. We did not know until we did those experiments where measles actually targeted the primary target cells of measles.
So you do the ferret experiment to figure out where to look.
Yeah.
Then you do the initial macaque experiment with the intratracheal inoculation to make sure that it works the same way in the macaque that it did in the ferret. And then you can move further into the macaque model, because then you've justified the investment in that model, and you can follow through to there. So you go from one model through to a more human-like, measles-like model.
But because it's simply not trivial to do these experiments.
But in a macaque, have you ever just aerosolized virus?
Yeah.
Where does it initially replicate?
So that was what we did once we then were able to do all of this experiment. Right. We were interested in really understanding, as I always see it, measles is a really great system because you've got both sides of a coin. You've got the attenuation side, you've got a great vaccine that really works, and you've got a really, really big disease, you can understand the pathogenesis. So you really can understand both sides of that coin. So no one knew where were the primary target cells. So the way to answer that question is exactly what you said. Aerosolize, give every single cell throughout the entire respiratory tract an equal opportunity to pick up measles virus.
I'm dying to know the answer. It's 10 minutes since I asked
lung.
you.
So in the deep lung, the
It took him years to do this
It took us years
experiment.
to do it. So in the
You
a
are gonna
primate
listen
model.
to what it took him to
But what
get
you have to
there.
understand, is where you really are looking, because the assumption, all those textbooks, right, draw people away
I wrote the
from.
textbook, yeah.
And it says epithelial cells. It says epithelial cells. People imagine it's a respiratory virus. The epithelial cells don't have that primary receptor.
Okay.
So right in the deep lung, there are alveolar macrophages, dendritic cells. And whenever we dialed back from the peak of infections, so day 7, day 6, 5, 4, 3, 2, we got back as far as day 2, you take the whole lungs and we inflate the whole lungs and then section the lungs, and literally you can look through the entire lungs, and because of the fluorescence, it allows you to identify those cells in the deep lung. And then what happens from there, day 2 infection, day 3, 4, there are small bits of tertiary lymphoid tissue in the lungs. And from the, this is BALT, bronchus-associated lymphoid
right.
tissue, and those are perfect for measles. If you're a lymphotropic virus, that's where you want to find. So you're picked up by an alveolar macrophage, you find this little, in inverted commas, this little BALT. That is a perfect place you to replicate. You've got all of this lymphoid tissue. From there, you go to the tracheobronchial lymph node. If the route of infection is aerosol, from the tracheobronchial lymph node, you just go systemic. Systemic infection gets you back to the upper respiratory tract, because remember, the next thing that you're trying to do with measles is be transmitted.
Yeah.
And then you have a problem. You have this lymphotropic life cycle where you've always used cells of the immune system, but you are a respiratory virus. So how do you get out? And this was really just worked out in the last couple of years when Chris Richardson and Roberto Catania, was he talking about PVRL4, the poliovirus-like receptor
Yeah,
4?
he
He talked
did.
a little bit
Yeah.
about that. So they at the same time identified this poliovirus-like receptor, PVRL4 molecule, which sits on the basolateral surface of the epithelium and in the adherence junction. And once the immune cell comes into contact with that adherence junction protein, PVRL4, that acts as the route from the immune system into the epithelium, and from the epithelium it goes to the apical surface and then gets spread.
So
I mean, it's beautiful.
where in the lung is this happening? Is it
Well, this is happening now
Upper?
in the upper respiratory tract.
Right.
So it's gone the whole way systemic, subepithelial cells, immune cells, coming into contact with the adherence junction in the basolateral surface of the epithelial cells, and then into the epithelial cells, and then the glycoproteins are sorted to the apical surface, and then the virus is released. I mean, it's beautiful.
So this whole systemic
Just
infection
beautiful.
is just a way for measles virus to get from the bottom of the lung to the top.
Yeah, it's so cool.
kind of circuitous?
It's
isn't it?
beautiful. I mean, it's when you see it, when you see pathogenesis, and when you see that development of initial target cells, amplification, further amplification, systemic spread. We just see the rash. You think of measles, you think of red spots. But what it has done before to get the whole way around, to get up into that, and then you can see in the nasal concha, and you can see in the trachea these beautiful epithelial cells all infected, ciliated bronchial epithelial cells.
Simultaneously elegant and convoluted.
It-
Very viral.
Yeah, it is. It's but does it work? Yes. It's
Yeah.
incredibly infectious. And can that virus get very effectively from the Philippines to New York or wherever it is? Absolutely.
So in your infected macaques,
Yep.
if you look later in an infection, when you're getting shedding in the upper tract.
Yes.
Do you see any infection of epithelium in the lower tract at all, or is it not bothering to infect there because it can't be transmitted?
It's still present in the respiratory
So
system,
it's
yes.
throughout the tract,
But
right?
it's heavily
Okay.
infecting in the nasal concha.
Do you know
It's just
why it's more heavily infected in
littered.
the upper tract?
I don't know. The issue probably is, I guess there's an awful lot more about this adherence junction PVR4 protein that we really need to understand. It might be a density of receptors. It might just be the physical. It's easier to get out of that. There are less layers of cells. There could be multiple of just simple
the microbiota.
physical reasons
Microbiota.
why.
Microbiota.
There you
And
go.
then, and then you have to think back to, and again, you see the lovely parallels as you're talking about the gut, and I'm thinking, now I wonder what's happening there with the mucus.
Do a sabbatical in her lab,
I tell you,
sit on our patio.
that patio is full.
Pretty full.
That patio is full.
Is it big?
No,
No, okay.
it's like a 3 or 4 seater.
But you see,
Could
the
you,
other
could
thing,
you
rotate
aerosolize
people
feces
in.
from it?
You can aerosolize species from anywhere.
That's true.
but the reason why that's important to understand is there's a project in the World Health Organization. They're trying to talk about aerosol vaccine delivery.
Really?
Now, if you don't really understand primary pathogenesis, you don't know whether or not, as you first asked the question, should I be delivering it to the upper? You could spend all of your time developing a beautiful project with the assumption that measles is a respiratory virus and you just dab it on its nose and away you go. And no, without understanding
the wrong place.
primary pathogenesis, you cannot do any sort of proper translational work, which is develop a vaccine which might be very thermostable, could be a powdered vaccine like this cool company Active Dry are working on.
right.
But they need to know where to deliver that. And if you put it up here, not a good place to go.
So they want to do this to avoid the injection.
Yeah, this would be
Right.
really very attractive. So again,
Like flu-ness, but for measles.
yeah, where polio leads, we follow. So you're going to be eradicated, we're going to be eradicated. The question is what will happen after post-eradication? Would you ever stop vaccinating? Should you ever stop vaccinating against measles? And then that links to where you started, and this is where you come back to CDV, and you come back to zoonotic infections, and you come back, because you know what? Give me measles vaccine any day. Compared to CDV, I would not want to have CDV.
Does it infect people?
Well,
CDV?
has it been used to vaccinate people against measles? Yes.
No, but I'm wondering in the wild, do humans ever acquire CDV?
Are there zoonotic infections?
Well, the
People
thing
with
at the
CDV?
minute is where it would be, it certainly would not happen. If it happened, it would not be able to be seen here. You would have to have huge swathes of people who are not vaccinated.
Okay.
But remember that the interesting thing about these morbilliviruses is they're very, very close family. So there's cross-protective T-cell epitopes. So what I know is if I get measles, CDV is a very, very safe virus for me to work with because I am not going to get infected with CDV. Can I? Because of the T-cell immunity.
So you're worried if you eradicate measles. So do you think we can eradicate measles at some point in the future?
It's really interesting. People are not as convinced that it's possible. It is as possible as for some of the other viruses. The people really disagree about that. I think that it's
People disagree about polio too.
Yeah. I think polio is, I think, might be even harder because it's a pretty stable virus that kicks around in the environment
it's in sewage,
for
whereas
quite a
measles
long
is gonna
period
be
of time.
person to person.
Exactly, and measles is much more labile.
And we use a vaccine for polio that back mutates.
Yeah, that's
the
the
big
issue. So
problem,
poliovirus might be slightly slightly harder than measles virus. The issue is with people not taking the vaccine at the moment, and this anti-vaccine.
Okay, so that is a
Nonsense.
But assuming, all right, let's assume we eradicate one day. Are you worried if we stop immunizing, then these other morbilliviruses could start infecting people?
Well, what do you know about a virus? What do you know about viruses? They
They
evolve
change.
to fill a niche.
Yep.
So I think I would like to know what other morbilliviruses are out there. And I think I would like to know what are the barriers to cross-species infection. I think that's incredibly important because, as I say, give me measles vaccine much faster than measles, and give me measles vaccine and measles much faster than CDV, because I've seen what that can do to the brain of a ferret, and I don't want that anywhere near my brain.
Well, and we've seen with smallpox, right? Aren't there pox viruses that appear to have
So there are
come in to fill
monkeypox
the niche?
infections are coming in.
And
Nobody
what's
got
the- yeah.
nobody got monkeypox
Yeah.
when everybody was
Yeah.
vaccinated
They
against
cross-
smallpox.
Exactly.
yeah.
Now we stopped vaccinating against smallpox, and we have monkeypox coming
See,
in.
you have to really think about this. No generation has lived, been vaccinated, and got the whole way up to 80 or 90 years old and died vaccinated yet. Vaccination is quite a big experiment. Is that correct? Certainly for measles.
Well,
not
for
for
smallpox,
polio.
it's not.
Smallpox, it's not.
Smallpox, we have generations
So
and
if you're
generations.
looking at what you're learning from monkeypox could be applied to poliovirus, could be applied
Sure.
to measles. We sort of say lifelong immunity, and we hope that that's the case. And I'm sure probably is more than likely is going to be the case. But don't be under any illusion. That's something we don't have the data for that, so we can't. We don't know that. As yet.
One of the main reasons of eradication programs is to stop immunizing at some point. You don't want to immunize forever, right? So if we can eradicate polio, the goal is to stop. Measles would be the same thing.
What do you think will happen with polio? Do you think
So
people
polio is
will
a very
stop?
well. Polio is a problem because the circulating vaccine-derived viruses are there, and if
Yeah.
we switch to inactivated vaccine, which is the plan, the guts will not be immunized.
It's
So
the
those-
new plan.
So the new plan.
That wasn't the original plan.
It's the new plan, and those viruses will continue to circulate.
Yeah.
This is what happened in Israel. They found wild viruses in the sewage. No cases of polio because everyone was immunized with
Yeah.
IPV, but the gut is susceptible. So I have this vision that we have to use IPV forever to protect us against the vaccine that we use to eradicate infection. However, even beyond that, there might be picornaviruses out there that could move in. There are certainly Coxsackies that are neurotropic that could evolve.
And what's
to
the one in
eliminate-
there's something in California, some
Well, there
patro-
was an outbreak of a high reporting of acute flaccid paralysis, which is a syndrome that can be caused by many things besides viruses. It's been associated with enteroviruses, but it's not at all clear that
So that's not
those
clear
cases
yet?
were caused by a virus at all.
Okay.
Yeah. But I think in all cases when you eradicate and stop immunizing, you have to wonder if there are other viruses out there that can substitute. And I think, as you said, we don't have enough information for most viruses to be able to answer the question. It's a big experiment.
And that's the sort of information I would like to collect. I think that that's important to know because we really will have to get to the point where those people, it would be so attractive to take measles out of the cold chain. That would be great
Yeah.
if that was possible. But there are a number of reasons, zoonotic infections, and I mean, heaven forbid, you can make the virus. I don't think that I think eradication is in some ways a redundant concept in the world of reverse genetics. It doesn't cost. It would cost what, $10,000 to make a clone. It's not that.
Well,
I
this
mean,
is the ongoing debate about
And that
whether
is something
to
that-
destroy the
Yeah.
smallpox
Yeah.
stocks
Yeah.
because you could make them again anyway.
Yeah.
Why bother to destroy
why
them?
bother to
Yeah.
destroy them?
a good
Because
question.
it's the same as measles. Should we eradicate it or should we get rid of the stocks? Because you can recreate that for
Polio is
next
very
to
easy.
nothing in the lab.
7,400
And you know
bases.
what? That's an incredibly infectious agent. And I'm not, again, sure that the world would want to be morbillivirus naive. Those are questions for people with bigger brains than me.
Unfortunately, I think they're decisions being made by people who don't necessarily have bigger brains.
Paul,
I wouldn't like to have to make those decisions.
I want to ask you one last thing. So your student Connor just defended, right?
He did.
He was working in Belfast. He didn't want to come here and join you in Boston.
He didn't. We didn't think that was a good idea. So I went back to Belfast not last Friday, but the Friday before, and he defended his thesis 4 hours.
4 hours.
Hours. Shower. Towel.
I can't understand his accent. I'm sorry.
I didn't think I had an accent.
No, you don't.
They're going to do subtitles on this, right?
All right. Well, congratulations, Connor.
Yeah, that's good.
He
He's
might
now
be
a
listening.
hepatitis C.
He's going to Glasgow, right?
Hepatitis C person with John McLaughlin
Right. Good.
in Glasgow.
Good luck. He's a TWiV fan. I want to do some picks. What do you think?
Sure.
To wrap it up, what do you have today? Pick of the week.
What I have, let me scroll down. We have these show notes in Google Docs, which we paste in letters to the show. So if you've sent a letter to TWiV, it's somewhere in this massive, massive set that I'm scrolling.
Does it have a page? Is there a page?
Oh, here we go. Yes. So my pick this week is it's an article on a site called Persephone Magazine. It's called "I Will Not Follow the Herd," and I found this absolutely wonderful. It's by somebody writing under the name Susan, which I think is a pseudonym for reasons that will be obvious once you read this article. It will probably offend people who you don't want to use your real name in offending, but it's about making intelligent decisions for your children. And how, after searching through Google for quite a while, she's realized that she made a lot of mistakes with her first child, and she's not going to make the same mistakes with her second child in buying into all of the PR and hype that you get from the big safety industry, and the dangers of things like seat belts and other things that we've been sold. You may notice some similarities between the arguments made here and arguments you may have heard elsewhere. I'll just leave it at that.
It's great. Yeah, highly recommended.
So,
short. You could read it quickly.
Yeah, it's about a 2-minute read.
All right, my pick is very self-reflexive. It's ASM Live, which is what we're doing right now. But there are also other sessions at ASM Live. Besides TWiV this morning, there were 2 by Stan Malloy, who was standing over there before. And there will be 2 tomorrow morning and 2 the following day, I believe. So they're really nice. They are streamed live. And the way the streaming works is if you come in halfway, you could actually start from the beginning. So it's pretty neat. So check out ASMLiveMicrobeworld.org.
So it's like TWiVo.
Yes, right. Exactly. I got that one. I got it. It is. It's a DVR on the microbeworld.org website. I have a listener pick from Neil. This is pretty cool. Dear TWiV, I noticed the listener pick animation of DNA replication from Marshall in TWiV number 278 and thought you would appreciate some more animations of this kind. The DNA animation is the work of biomedical animator Drew Berry. I believe that you read a letter discussing his work on TWiP number 24. You can see more of his work on the YouTube channel WeHi Movies. WeHi is the Walter and Eliza Hall Institute of Medical Research in Melbourne. And he gives us a link to the YouTube channel. Very cool animations there. For the past year, Drew has been working with 3 trainee animators as part of a visualization project called VisiB+. Their animations, The Hungry Microbiome, Cancer is Not One Disease, and Inflammation and Type 2 Diabetes have just been released and are available at the VisiB+ website, which he gives a link to. I hope you agree that these are both scientifically informative and visually very striking. They're gorgeous animations.
These are cool.
Looking at the one where they have a molecule of DNA and it's being bound by histones and wrapped up.
Yeah.
It's just gorgeous. These science
We're not on
animations.
Skype, so I can actually watch the YouTube video while we're doing
Yeah,
this.
it's not going to play. Oh, there you go.
Yeah, I don't have the sound on, so you
Very
can't
nice.
hear it.
You guys don't want to pick anything, do you?
I actually would like to say something about that. It's a tweet from earlier on this week, and since you've asked, there's a really, that I'm passionate about vaccinations and measles vaccination in particular. There is a really, really great movie made by a high school film club in Carlsbad, California. They basically made this movie called Invisible Threat. Do you know this?
Yes, I was.
And this is something which really
going to be a future
deserves,
pick.
honestly, well, this is something that deserves
Just scooped his pick.
our support as far as a little bit of publicity on TWiV. These kids have done a tremendous job. I was in touch with their teacher this week, Lisa Possard, and she sent me the full. She's sending me the full movie, but they have very carefully, very methodically, and incredibly professionally put together a movie called Invisible Threat about the importance of vaccination, and they interviewed people, and I think it's a real credit to them. They're getting some flack.
Of course, from the people you would expect,
lobby,
yeah.
from the usual suspects, and I think attacking a group of high school students just because they have gone and looked at the evidence and made their own decision that vaccines do not cause autism, that this is all blown up out of all proportion. I give them great credit, so I think it would be wonderful if you could post that pic and put some sort of a link to this group of people. This would be great.
So you did tweet it to me last week. And said,
I tweeted it,
this,
and I thought to myself, if I get the chance today, I'm going to give them a little bit of a plug.
How could you not have a chance?
Well, you never know. Have to keep an eye on
you're not
you.
a guy that doesn't, well, never mind. Julie, you want to pick anything?
I'll find something to send you.
You'll find something, no problem. We don't want to put you
It
on
will
the
not be a
spot.
A link to be
biological.
disclosed later.
Patio, something from the patio.
It's going to be a box.
Oh, nice, really nice.
Picture of the patio.
On this patio, do you have drinks?
Yeah.
What do you drink? What do you drink on the patio?
What do we drink?
Beer, wine, margaritas? What's your favorite?
Used to be red wine. We recently switched to white. Laura's doing an experiment.
Ah, I see. I can imagine that experiment. All right, that's TWiV 286. You can find it at twiv.tv, also on iTunes. And if you like it, we don't charge you money for it, but please go to iTunes and subscribe. It's free. And make a comment or leave some stars. That helps to keep us visible so more people find us. We're really lucky to have lots and lots of subscribers and great fans, but we want more. And so please do that. And if you have any questions and comments, we usually read them on the show.
Eventually.
Eventually. We will get to every single one of them, except the ones where Alan tells me not to read them. Like halfway through the first sentence, right? You've done that before.
No, there are only a couple of
A few of those.
people
You could,
who've written repeat letters on the same subject.
we know who they are.
Yes.
Anyway, you can send your letters, email to twiv@twiv.tv, and we'll be happy to read them. I want to thank our guests today for joining us. Paul Duprex from Boston University. Thanks.
Very good to be back.
It's fun to have you, and I hope you enjoyed your second time. Come back, okay?
Well, you can come back. You should be coming back to us. So maybe whenever we start to open NEIDL, you'll come and do a TWiV from that. Huh?
We'd be happy to always. Alan Dove.
If we have a symposium to begin with, let's
That would
put
be
a
great.
TWiV in the symposium, huh?
All right.
That would be great.
Yeah.
Julie Pfeiffer from a backyard in Dallas somewhere. Southwestern University in Dallas. Thank you so much for joining.
Thanks for having me.
today.
It's fun.
Appreciate it. You should come back too as well when you figure out this gut business with
Yeah.
polio. Okay, you can tell us about it. And Alan Dove, thank you.
Thank you. Always a pleasure.
Alan Dove is on Twitter, and he's also
Right now.
Mine died. I don't know what happened. Did we get any questions that we should
got,
answer?
there are some, careful aerosolizing feces sounds like dual use. Very good point. Yes. I could probably think of more than two uses.
Oh, it's great to finish on a high point.
Oh, here's a good one. Given pro and anti-virulence interactions between viruses and bacteria, why so little virology at ASM meetings?
Well, that's a topic
Wow.
for an entire show, right?
We'll
That's
let
a
other
whole
people
other
handle
thing.
that question.
Yeah, that's buried in history. Alan Dove is at turbidplaque.com. That's his blog. I'm Vincent Racaniello. You can find me at virology.ws, soon to be in a backyard in Dallas, I guess. I want to thank ASM for letting us do this here at the general meeting. We really appreciate it. And my phone stopped buzzing. Is that your phone?
It's my phone buzzing.
You.
My mother.
And I particularly want to, your mother is see if it's actually your mother. Yeah? It's not?
No, it's not.
Okay. I thanked ASM. I want to thank the people behind the scenes. Warren, thank you very much. Ray, the camera guys, and Chris Kandian, our invisible producer. Thank you, Chris. Yeah, you're welcome. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shock.
It's an unusual room. Never seen anything like this.
Anybody who has access to this facility first has to go through an R scan.
HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right?
Remember, this building is basically a second building inside the main building.